Swayam Prabha. Digital India. Educated India.
Hi everybody, this is Dr. Ravindra Mardi, Associate Professor in the Department of Biochemistry, Kasturba Medical College, Manipal. In this lecture, I will be discussing about the structural organization of proteins as a basis for understanding its function. I will be giving emphasis on hemoglobin and collagen structure. Before moving into how the hemoglobin and collagen structures help in their functions, we will look into the different structural organization of proteins, how the proteins are organized. We have different structures of proteins. In that one is the primary structure of the protein. Primary structure of protein is nothing but is a linear chain of amino acids joined by one another by the peptide linkages. On one side we have a amino group, then other side we have carboxyl group. In between the amino acids are linked together by peptide linkage. Peptide linkage is formed by a amino group of one amino acid with the carboxyl group of another amino acid with the release of a water molecule, and this peptide linkage is a very strong linkage. It is a covalent linkage bond formed, and it is not you can't denature the when the proteins are undergo denaturation, the peptide linkages are not broken down. So this is a strong linkage, that is the primary structure. We also have the secondary structure. The primary structure is wound or bent like alpha helical form. We call it as a alpha helix, and it can be seen in case of beta sheet like structure. It may be anti-parallel or parallel beta sheets. These are due to the hydrogen linkages, hydrogen bonds in between the amino acids. They cause these different secondary structures. We also have some super secondary structures like alpha loop alpha structure. This is alpha, it is a loop and alpha structure, beta alpha beta structure, beta meander. So there are different super secondary structures are also available. Whereas tertiary structure, here it is a single polypeptide chain. It is a further three-dimensional organization of the proteins. So then the tertiary structure is there. It has the primary structure, then it has a secondary structures, and multiple. It gives a three-dimensional structures. For example, like myoglobin. Okay, that gives the tertiary structure. If there are many polypeptide chains are interacting to each other, and they form a quaternary structure. Example is hemoglobin. Okay, these are the four basic protein structures we saw: primary, secondary, tertiary, and quaternary structure. Let's move on to the how the structure of hemoglobin helps its function. We'll see. Hemoglobin is a molecule that is present in the RBCs. It helps in carrying the oxygen. So it is made up of two parts, heme and globin part. Globin is the polypeptide chain that is made up of amino acids, and heme is a protoporphyrin ring with iron in the ferrous form at the center. We have different protoporphyrin 9 rings, and then iron is present, and this iron in ferrous form, it forms a coordinated complex with a nitrogen, 4 nitrogen of the protoporphyrin ring, and 2 bonds with proximal histidine F8, and one is attached to the oxygen and then to the distal histidine E7. This is the structure of the heme. And what about hemoglobin? Hemoglobin, take for example hemoglobin A, it is made up of 4 polypeptide chains, alpha 1, alpha 2, beta 1 and beta 2. two alpha and two beta chains are there, and these alpha and beta chains are held together by strong hydrophobic bonds. These two work together as a unit, alpha 1 beta 1 and alpha 2 beta 2, because of the strong hydrophobic bonds in between these alpha and beta chains. What happens? The two dimers are held together by weak ionic and hydrogen bonds. These two bonds are strong linkages, hydrophobic bonds, whereas the dimers alpha 1 beta 1 alpha 2 beta 2 are linked by weak linkages, weak ionic or hydrogen bonds. So, the hemoglobin is present in two forms, one is T or taut form and another is R or relaxed form. In the T form, it is deoxyhemoglobin form. Oxygen is not attached in the T form because it has got a low oxygen affinity in the T form, whereas once the oxygen molecule binds to this T form one molecule, then there will be breaking of these weak bonds, weak hydrogen ionic bonds are broken down. We call it as salt bridges. They are broken down, and that helps changes a conformational change in the hemoglobin, and it forms a relaxed form, and relaxed form has got high affinity for the oxygen. In a cooperative manner, the binding of first oxygen is little bit difficult, but further the oxygen binding is more easily, it is bound to the hemoglobin in the R form. So, when you see at the oxygen dissociation curve, in the x axis it is partial pressure of oxygen, in the y axis is the percent saturation of the oxygen. If you see at the 100% PO2 level, that is at the lungs, for almost all hemoglobin is 100% saturated. When the PO2 drops down in the tissue level around 40, it releases the oxygen to the tissues. So, it is bound completely in the lungs, and then it is given back to the tissues at the partial pressure of oxygen decreases. So, how it helps, how that oxygen binding helps in this cooperative binding, and what are the changes it causes when the binding of the first oxygen to this histidine of F8 histidine, it binds to the iron of that histidine, and it pulls that iron, pulls the iron molecule which has bound to the distal histidine F8, and then it pulls that iron into the plane, porphyrin plane, and this causes the breaking of the salt bridges, and that will change a conformational change in the hemoglobin molecule, and more of it converts T form into R form, and there is a high affinity for the oxygen. That is how this structure helps in its function, that is binding of the oxygen. So, this proximal histidine and other residues, they move into the plane of porphyrin ring and breaking the salt bridges and converting that T form of hemoglobin into R form. Once the oxygen is delivered, it goes back into the T form. That is about this hemoglobin, and then we look at the structure of the collagen, how the collagen structure helps its function. The basic function of collagen, it gives it is a fibrous protein, it gives a tensile strength to the tendons, or it may be present in different tissues in the gel matrix. So, what is the structure? It is basically it is a alpha helical structure, 3 polypeptide chains. It is a triple helical structure, 3 polypeptide chains are wound against each other. So, in this polypeptide chain, the majority of the amino acids are glycine, proline and lysine. So, glycine, proline and lysine are the amino acids, around 1000 amino acids if are there. They are repeated sequences of these amino acids. These amino acid structures play a very important role in the function of this collagen. What happens? The proline Proline, it has got a ring structure which helps in the causes the bends or kinks in the polypeptide chain. When the polypeptide chain is bent, there the proline molecules will be coming here. So, to give a kink or bend to the polypeptide chain, whereas glycine, it's the smallest amino acid, it fits in between the restricted places. When these polypeptide chains are wound against each other in the alpha helical form, that winding part there is a restricted place, and that is occupied by the glycine. And furthermore, these proline and glycines are undergo hydroxylation by the enzymes hydroxylases, and those gives the hydroxyl groups the proline and lysine, and these hydroxyl groups they form bind against each other, and that hydrogen bonds are formed. And then once this collagen structure is formed, there will be cross linking of these fibers, collagen fibers, with the help of the enzyme lysyl oxidase. Overall, the function, the structure of this will help in its function in this collagen structure. To summarize, we discussed about the different structural organization of proteins. We saw primary structure, which is primarily held by the peptide bonds. Then we have secondary structures, alpha helix, beta sheets, and we saw different super secondary structures also. And then tertiary structure is a polypeptide chain with three-dimensional structure. Example is myoglobin. And then quaternary structures, there are many polypeptide chains interacting to each other, like example is hemoglobin. So, in hemoglobin structure, we saw what is heme and what is globin and how it is heme is formed, that protoporphyrin 9 and that iron molecule in the ferrous form. And then we saw what are the T form and R forms of hemoglobin, how they help that weak bonds, hydrophobic bonds, strong bonds, hydrophobic bonds between alpha and beta chains, and in between the domains are weak ionic hydrogen bonds are present, and those bonds can be destabilized when the oxygen binds by pulling the ferrous ion into the plane of porphyrin ring and destroying some of the salt bridges and converting T form into R form and help in binding of the oxygen to the hemoglobin. We also saw the oxygen dissociation curve also, how it helps in binding at the high oxygen tension in the lungs and release of the same oxygen in the tissue level. Next, we discussed about collagen structure. It is a alpha helical structure, 3 polypeptide chains, around 1000 amino acids, and majority of them are glycine, proline and lysine, and the role of proline played helps in the giving a kink or bend to the collagen, and the glycine being the smallest amino acid, it sits at the restricted places, and then there is a hydroxylation between proline and lysine to form hydrogen bonds, and the fibrils are joined together by this cross linking of fibrils takes place by lysyl oxidase. I hope this lecture will help you to understand this different structural organization and its how it plays its role in functioning of that molecules. Thank you.